All right, so today we are going to continue our discussion of Chapter 3. It's a big one, right? I mean, it's our third lecture on it. It's a lot of material in Chapter 3. It's about metabolism. And metabolism is so important for every organism, especially for microorganisms, because it's how an organism gets its energy by breaking down substances, and then also how it builds up substances, right, that it needs for its cells to work. So up until now, we have talked about basically the catabolic stages of metabolism, or catabolism, breakdown, and we're going to finish that up today. And then we're going to cover the anabolic part, so anabolism, building up or synthesizing molecules. Did you guys notice? Probably not, because it just went live. There is a new quiz. Okay, so at 12:30 today it was set to start. You have from today until Friday at 5, just like always, to do it. It's going to cover the last part of Chapter 3, which we cover today, and then it's also going to include the first part of Chapter 6, which we'll cover on Thursday. And then next week's quiz, which will go live next Tuesday and close next Friday, will cover the remainder of Chapter 6. Look at me. I'm organized, thinking ahead for the first time this semester. All right, so we'll go ahead and talk for an hour, finish up the material from Chapter 3, and then the last 15 minutes of class, we'll go over some of the most missed questions on the exam, okay? So as I said, we covered a lot of catabolism already, right? Breaking down some sort of substance in order to make energy. That's what catabolism boils down to, okay? We already covered the most beneficial way by which organisms do that, and that is through which pathway? Oxidative phosphorylation, or more specifically, aerobic cellular respiration, because we'll learn today that oxidative phosphorylation can actually be done in anaerobic conditions as well. So what we've covered so far is we've gone through the process of glycolysis, followed by citric acid cycle, and then into oxidative phosphorylation using an electron transport chain and an enzyme ATP synthase. Remember that? That is very energetically favorable for the cell. The cell can make a ton of energy in the form of ATP using that pathway. And that pathway, again, is aerobic cellular respiration, okay? We also talked about a deviation from that, where you go through glycolysis, and then if there's no oxygen available, the pyruvate can be funneled into, you guys know, fermentation, right? That's one option. So we talked about fermentation pathways. Are these as energetically favorable for organisms? No, not by a long shot. But some organisms don't have a choice, and that's what they do, right? So we talked about lactic acid or lactate fermentation done by bacteria, done by our muscle cells. And then we talked about alcohol fermentation. So today what we're going to do is we're going to now talk about anaerobic cellular respiration. Okay, and you're going to see a lot of the same things that we saw last time. You're going to see a very familiar electron transport chain and a very familiar ATP synthase enzyme. But what you're not going to see is what? Oxygen, right? Anaerobic without oxygen. So we're going to go through anaerobic respiration now. We're still in catabolism, guys. We are still breaking down things, molecules, into basically releasing electrons rather, and making energy. So we have to remember that respiration can occur with oxygen under oxic conditions, or in anoxic or without oxygen conditions, right? And sometimes students get confused because they see this term oxidative phosphorylation, right? That's the term we use to describe making ATP using electron transport chain and ATP synthase and proton motive force and all that. So sometimes students say, oh, oxidative uses oxygen, but that's not true. What does oxidative mean in terms of chemical reaction? Loss of electrons, right? Redox reactions, the oxidation part is losing electrons. So that's what it's referring to here. The loss of electrons from a molecule, those electrons ultimately get transferred to the ETC and cashed in for ATP, right? So don't let this term confuse you. Oxidative phosphorylation can be either aerobic or anaerobic, oxic or anoxic in terms of the conditions. Okay. So of course, aerobic respiration requires oxygen, right? That's what aerobic means. So these are organisms like you and me that breathe oxygen and use oxygen and require oxygen for our life. But there are many organisms, many microorganisms, that do not require oxygen and may even be harmed with it. And that's where we get anaerobic respiration. So for aerobic respiration, the oxygen comes in because it acts as the final electron acceptor at the very end of the ETC. Does that mean oxygen has a high reduction potential or a low reduction potential? If it's at the very end of the ETC, the very last electron acceptor. Huh? High. Yeah, it's got to have a high reduction potential, a high potential to be reduced and to gain electrons. So it's pulling those electrons toward it with every step, right? And those electrons wind up with it. So it has a high potential to be reduced. But with anaerobic respiration, no oxygen, but we still have to have a final electron acceptor. We still have to have a molecule at the end of the ETC with a really high reduction potential that serves as the endpoint for where the electrons wind up at the end of this process. It's just not oxygen. It's something else. And it could be any number of things. We'll see some examples of what they are, nitrate, sulfate, something like that. And we have to remember, too, you may be like, well, why does anaerobic respiration even happen? Well, a lot of organisms are anaerobic, right? The first organisms were anaerobic because the early Earth had no oxygen, right? So this anaerobic respiration, which is a lot more beneficial than fermentation in terms of the amount of energy you make, had to have been around for a long time. So fermentation is also anaerobic, no oxygen, right? But you don't have oxidative phosphorylation either. You don't have any kind of external electron acceptor. There's no ETC. There's no ATP synthase, right? You immediately go from glycolysis, and then you regenerate that NAD+, and that's done, right? Boom, boom, fermentation done. So it's not going to be nearly as beneficial to the organism in terms of how much energy you make. Instead, it uses what's called substrate level phosphorylation, and that's a more direct way of making ATP. Instead of using an indirect electron transfer, proton motive force, and then ATP synthase, it just makes ATP from ADP. So just as a review, the two main types of synthesis of ATP that we've talked about so far are your oxidative phosphorylation and your substrate level phosphorylation. Make sure you're clear in your mind what these mean, be able to describe them, the differences, the similarities, compare and contrast. That sounds like a really good short answer question. Maybe which processes, which pathways uses which of them would be good, right? So oxidative phosphorylation, again, is making ATP, but making it more indirect, right? So you have electrons being transferred from carrier to carrier into the electron transport chain, where a proton motive force or hydrogen ion gradients form. And then that ultimately makes ATP thanks to the enzyme ATP synthase. And that's oxidative phosphorylation. Substrate level phosphorylation, on the other hand, is a direct way to make ATP from a DP and an inorganic phosphate. And it does not require the transfer of electrons, nor does it require a proton motive force, nor does it require an ETC, nor does it require ATP synthase, right? It's a much more direct way, and we see it in glycolysis. Whenever ADP gets phosphorylated, we see it in the citric acid cycle whenever ADP gets phosphorylated. So the one that's going to generate the most energy, of course, is oxidative phosphorylation, because you're using the energy of a PMF, right? You're using a lot of energy that's been stored in those electrons. You get a much smaller amount from substrate level phosphorylation in glycolysis and the citric. You guys clear on those two different processes?
Could you repeat the last thing you just said?
The last thing?
A lot less?
You get a lot more energy in oxidative phosphorylation because you're using all that energy that those electrons are carrying in that PMF. You get a lot less energy in substrate level phosphorylation, which is how you make ATP in glycolysis and the citric acid cycle. So this figure from your book just sort of breaks down the metabolic diversity that we see among microorganisms. So remember chemotropes, right, which need to use some sort of substance to get their carbon, right? They can't make it themselves. so they have to ingest it from a source. Can be broken down into chemoorganotrophs and chemolithotrophs. What's the difference? One uses organic carbons, and the other an inorganic substance, right? That's their source of electrons. Think of electrons or energy. Their source of electrons is going to come from either an organic substance, like sugars, for instance, or carbohydrates. That's the case of chemoorganotrophs. Or it's going to come from some inorganic substance, and that's the case of a lithotrope. And so some examples of energy sources or electron sources for chemolithotrophs include hydrogen gas, H2, hydrogen sulfide, iron, believe it or not, ammonium. Those are just some examples. Okay, so that's the starting point, right? Not glucose, like for chemoorganotrophs. They're not starting with glucose and glycolysis, right? They're starting with this inorganic substance for this process. Despite what they use as their starting point, though, the next step is the same, right? They get these electrons from some source, and then guess what happens to those electrons? They get sent to an electron transport chain. And then the electrons get transferred from protein to protein, hydrogen ions get transported, a PMF forms, and then ATP synthase makes ATP. All that's the same. Doesn't matter if you're a chemoorganotroph or a chemolithotroph. You still use the electrons that you got from whatever food source you're using, and you use that to make ATP. It's just the source, the initial source is different. And then, of course, the final electron acceptor will be different depending on whether it's aerobic or anaerobic, right? Respiration occurring. So if it's aerobic respiration for either a chemoorganotroph or a chemolithotroph, if it's aerobic, then oxygen will serve as that final electron acceptor. But if it's an anaerobic respiration pathway, if it's chemoorganotroph or chemolithotroph, oxygen will not serve as the final electron acceptor. You'll have something else there, which could be sulfur, sulfate, nitrate, it could be any number of things. But it's not going to be molecular oxygen, O2. Both can do with or without. All organisms use glycolysis. Another difference I do want to point out is up here. Notice the chemoorganotrophs that use an organic electron donor like sugars, glucose. They're the only ones that are going to go straight into fermentation. Chemolithotrophs do not take advantage of fermentation pathways. You guys see that? These guys go to fermentation. And then on the right-hand side, we have our phototrophs, right? And these are going to be the ones that don't need to ingest any kind of food to serve as their electron donor or their source of energy, right? They can make it themselves. So they're going to be able to make their own sugars through photosynthesis, and that's going to serve as the electrons that can then be used to make the chemical energy ATP for their purposes. So that's what's shown here. So sunlight is what they need to generate that electrons that then go through the electron transport chain and generate the PMF. And that can also happen with or without oxygen. Okay? So guys, terminology is super important. Okay? So make sure that you're able to use these terms of aerobic, anaerobic, chemoorganotroph, chemolithotroph, oxic, anoxic. You're able to use those terms correctly and understand what they mean. All right, so let's talk about E. coli then. Probably the best studied of all microorganisms. What about its respiration? What about its catabolic pathways? Well, E. coli is extremely versatile. What does that mean if something's versatile? Can do many different things in many different ways. So E. coli, yes, it's a chemoorganotroph, and it has electron transport. Okay, that's similar but not identical to Paracoccus D nitrificans. This is a genus and species of another spherical shaped bacterium, gram-negative, whereas E. coli is bacillus-shaped, rod-shaped. But these guys behave the same way, and they are extremely versatile and can make energy in lots of different ways. If E. coli has oxygen and needs oxygen, and it's in an aerobic environment, it can absolutely do aerobic respiration. If E. coli is in an anaerobic environment, it can participate in fermentation if there are no external electron acceptors available. But if it does have external electron acceptors available in an anaerobic environment, then it can do anaerobic respiration as well. It can do all of them. And that makes it very good at adapting to new situations and surviving in a variety of different environments. Now, which one do you think is going to be the preferred one? Absolutely aerobic. You make a lot more energy in aerobic cellular respiration than you do in the other two. What do you think comes second? Anaerobic, absolutely. And third, our friend fermentation, yes. So it can optimize under multiple conditions. If it has an organic carbon source, it'll grow the fastest using aerobic cellular respiration. And next, if it has a final electron acceptor other than oxygen available, it's going to choose to do nitrate respiration. That's a form of anaerobic respiration. But then if there's no nitrate or any other kind of cellular acceptor or electron acceptor available, then it will finally do fermentation. But the point is, it can do any of it and all of it, which is pretty cool. It also has a variety of different proteins that it can use in its electron transport chain, which is an evolutionary advantage. Because if it's not able to make a particular type of protein, or for whatever reason that protein's not functioning properly, it can just insert another one. So this figure from your book shows you E. coli's respiration pathways. Not the whole thing, just the ETC portion. And on the left, it has your aerobic respiration. This one's going to be preferred because you make more energy with aerobic cellular respiration. And on the right is an anaerobic respiration pathway, not fermentation, anaerobic respiration on the right. And this would be sort of second best. So if oxygen's available, then this is the pathway that's going to be used, the one on the left. You'll notice these big purple blobs are the proteins that make up the complexes in the electron transport chain. In this case, there are just 3 of them. In the previous examples, we saw 4. It differs, and E. coli has the ability to change out these proteins and use different ones. They just have to be able to transfer electrons and work with the quinones and work with the cytochromes and work with the, what are the other ones? Ubiquinone, coenzyme Q. They have to be able to exchange electrons with these different players, and then they can function in this way. What other thing do they have to be able to do, these proteins? They have to be able to exchange electrons. What is it? Protons. They have to be able to transport protons across the membrane. Remember how the ETC works? Electrons get transferred from protein to protein. And with each transfer, some of these complexes will move protons or hydrogen ions across the membrane and generate a hydrogen ion gradient or proton motive force. You guys remember that from last time? Okay, good. All right, so let's go through aerobic respiration on the left. Again, here's our lipid bilayer. This is happening in the cytoplasmic membrane. These purple blobs are our protein complexes. The electrons get dropped off by our electron carrier NADH, or in this case FADH2 for this complex. Once the electrons get transferred to the proteins in the ETC, hydrogen ions get transported across the membrane. You guys see that over here? I'll do it again. All right, here's our lipid bilayer of the plasma membrane. Purple blobs are our proteins that make up the ETC. So the electrons get dropped off by the electron carriers, NADH and FADH2. Once the electrons get transferred here, they move to the next complex, and a proton or hydrogen ion is going to get transported across the membrane. So you get 4 protons here with this first complex. The electrons move along. You get another 2 here, another 2 here, for a total of 8 protons or hydrogen ions on the extracellular side of the membrane. That's the outside of the membrane. So now we've got all of these protons here on the extracellular side of this membrane. But remember, they want to equilibrate. They want to have an even amount on both sides of the membrane. So what do they want to do? They want to come back down. Can they come back down on their own? No, they're charged, right? So ions, charged atoms, have to have a channel through which they can be transported. That's where ATP synthase would allow them to pass back through and ATP would be generated. And then notice our final electron acceptor where the electrons wind up at the end of this is oxygen, and that will make water as the byproduct. You guys see that? That's key. Circle it on your slides. That oxygen, that's the final electron acceptor, right? Where the electrons wind up and water gets made as a byproduct. Right, so that's preferable, right? You make a lot of energy. Look at all those hydrogen ions that got transported. All that gets cashed in for ATP energy. On the right, though, what if you don't have oxygen? Well, you can still do respiration and not have to do fermentation. Just use something else as your final electron acceptor. So in this case, nitrate will do the job. And so this process is called nitrate reduction. It's a form of anaerobic cellular respiration. So here, same thing happens, right? Electrons dropped off by the electron carriers, NADH and FADH2. Electrons get transferred from protein to protein in the ETC. Protons, or hydrogen ions, get transported across the membrane to the extracellular space. But there's a difference. We've transported fewer hydrogen ions. You'll see that? How many have been transported here? 6 as opposed to 8. So you transported fewer hydrogen ions. That means you're going to make fewer ATP molecules when ATP synthase passes these in for energy. Right? So this is a less productive pathway using nitrate down here as our final electron acceptor. But it's still better than fermentation.
So ATP actually.
That's right. When they pass back through, ATP synthase is a really cool enzyme. It has a domain that allows for a channel, and the hydrogen ions pass through it from the outside back to the inside. And that energy, as it goes through, generates it to rotate, and ADP gets phosphorylated to make ATP. Let's talk about the chemolithotropes for a minute. So these guys use inorganic chemicals to start the entire process as their electron donors, right? So for instance, hydrogen sulfide, H2S, hydrogen gas, H2, maybe even iron or ammonium. And so the process is going to begin with oxidation of this inorganic electron donor, just like with chemoorganotropes, it began with oxidation of that organic electron donor, glucose, right? So we're going to oxidize, break it down essentially, and let some electrons be taken from them, right? And those electrons that are taken from that electron donor enter the electron transport chain directly, okay? So in the case of these chemolithotropes like Ralstonia eutropha, a microorganism that is an aerobic chemolithotrope. Remember, chemolithotropes can be aerobic or anaerobic. This one uses oxygen as the final electron acceptor. So if we look at this figure here, gosh, it's hard to see with the light. Is that too dark? I should have made this figure bigger. Sorry, guys. But if you look here, right, we have the electron transport chain, 1, 2, 3, and then there's your ATP synthase at the end. So hydrogen, H2, serves as your electron donor, and it directly transfers electrons to the first protein in the complex, right? And then the electrons get transferred from protein to protein. The proton motive force gets made. The hydrogen ions pass back through the membrane through ATP synthase, and ATP gets made. And what serves as the final electron acceptor? Oxygen, because oxygen is used because it's aerobic, right? So differences here, we have hydrogen 2 as our electron donor. It directly gives its electrons to the ETC. The electrons get transferred. The final electron acceptor is oxygen. And ATP gets made from ATP synthase. Do y'all want them off or on? Off? Yes.
Is the reason why only 3 to 4 hydrogens get passed back through ATP synthase is because it needs it to make more H2 to start the process over again?
Exactly. Yep. Yeah, because see, 3 or 4 hydrogens go through here, but I think that just means at a time too. So they'll eventually, well, actually no, you know why it's 3 to 4? It's because it needs to be balanced. So here you have 6, and you have some here, and so you only want 3 or 4 to go through to make a balance inside and outside the membrane. That's a good point.
Just to clarify that NADH is not giving off electrons.
That's right. In this case, NADH does have a role, but it is to build cellular materials from these products. It's not into the donation of the electrons at the beginning of this pathway comes from H2. So both chemoorganotropes and chemolithotropes depend on oxidative phosphorylation to make their energy from their quote-unquote food, okay? And they can be aerobic or anaerobic. If they're aerobic, oxygen will be the final electron acceptor in the ETC. If they're anaerobic, it will be something else. The major difference, as I've already stated, but it's here in your notes, so it's really good to make sure it's written down somewhere. The chemoorganotropes are going to have to use organics for their carbon source to begin the whole process. Chemolithotropes are going to use an inorganic source. A lot of times it's CO2 is their inorganic source for these processes. Like autotrophs, right, photosynthetic creatures will use CO2 as the reactant to build products. And chemolithotropes are able to use a reverse electron transport system to form reducing power, which is kind of cool too. Maybe we'll see that in some upcoming chapters. So phototrophy, we can't leave those phototropes out. We talked about chemoorganotropes and chemolithotropes. We have to talk about phototropes too, because a lot of the microorganisms are autotrophs. They're able to generate their own chemical energy from sunlight, right? So these guys will use sunlight, generate a photon motive force. They also will use ATP synthase to make ATP, but it's through a process we call photophosphorylation. So we don't call it substrate level phosphorylation. We don't even call it oxidative phosphorylation. We give it its own name, photophosphorylation. And just as with the other ones, it can happen in oxygenic environments or anoxygenic environments. So these phototropes, if they use oxygen, then they ultimately form oxygen as a waste product. It's a waste product for them, but for us, we benefit from it, right? Think about plants that photosynthesize and release oxygen into the air, the same oxygen we breathe. So what are some oxygenic organisms? Those would be your cyanobacteria, your algae, your plants. These are all oxygenic phototropes. Then you have anoxygenic phototropes, which are many of the bacteria. They're believed to have evolved first and be the oldest, and they have more metabolic diversity. Again, the early Earth was anoxygenic, right? There was no oxygen there. So any of these anaerobic, anoxygenic species that we find, they've been around a really, really long time. And the others likely evolved from them. Purple bacteria, which is shown in this really cool picture here in a salt marsh, these guys are examples of anoxygenic phototropes that are common in aquatic environments without oxygen. So let's go through how a phototrope would go through this process. It's really cool. It's not all that different than what we've been doing, but it's got one important difference, and that is the presence of the photosynthetic reaction center. And you likely have heard this term before when you studied photosynthesis, maybe in a botany class or general biology. So basically, these photosynthetic reaction centers are embedded in the lipid bilayer of photosynthetic microorganisms, and they contain a chemical called chlorophyll, which is a green colored pigment. So basically, sunlight will come and strike the membranes of these cells, these photosynthetic organism cells, and they will hit the electrons in the chlorophylls of these reaction centers and excite those electrons, and now those electrons are full of energy. And the electrons will be transferred from chlorophyll to chlorophyll to chlorophyll, which is what these molecules are here, all the while building more and more energy within those molecules. And those electrons will be transferred to an electron transport center protein complex. You'll notice the cytochromes here. You'll notice the quinones, the Q cycle that we talked about last time. And the exchange of these electrons between the photosystem or the photosynthetic reaction center and the proteins in the electron transport chain will generate hydrogen ions to be transported across the membrane. Now you have a gradient of hydrogen ions. They're going to want to transport back across the membrane. So they use our friend ATP synthase to do that. So that ATP will be formed once these hydrogen ions that accumulated outside the cell will come back into the cell. So you still have electron transport, you still have oxidative phosphorylation. We just don't really call it that. We call it photo oxidation. Or what do we call it? Photo phosphorylation. Thank you. The term left me there for a minute. Photophosphorylation. But it is similar to oxidative phosphorylation. Any questions about that? I know this is a lot of information, but hopefully you guys are seeing similarities, figure to figure, process to process, right? And I recommend that when you're studying, you look for those similarities. It'll be like you're learning the same thing over and over again, right, with small differences here and there. And if you're able to assimilate that information and put it together and see how things are similar and different, it's going to really help you. All right, now we are switching gears. And we're going to talk about anabolism. What is anabolism? Building up, biosynthesizing large things from small things. So let's talk about how microorganisms do anabolic pathways in metabolism where they build the macromolecules. Remember, there are 4 classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And we need all of these, and microorganisms need all of these, right, in order to do their cellular functions. So there has to be a way by which these organisms build these molecules. So again, anabolism is the biosynthesis of cellular macromolecules. In order to do biosynthesis, microorganisms need certain elements. Okay, I mentioned carbon and nitrogen here because those are the first 2 we'll talk about. But they don't just need carbon and nitrogen, right? They're going to need oxygen and hydrogen and phosphorus and sulfur, all these very important elements to build their macromolecules. So where do they get them from? Well, when it comes to carbon and nitrogen, they actually get most of those from the atmosphere because there's quite a bit of CO2 and nitrogen gas in our atmosphere. The problem is it's not in what's called a usable form, okay? The CO2 is, the carbon is sort of locked up in some chemical bonds. The nitrogen, think about an N2 molecule. It's actually a covalent triple bond between the 2 nitrogens. That's tied up tight. That's hard to get those nitrogens free. So these elements are essential for the cells to be able to build macromolecules. And while there's a lot of them around, they're not in the right form to be used. So microorganisms, a lot of microorganisms, have the ability to transform these CO2 and N2 molecules in the atmosphere into usable forms. And we call that fixation. So you may have heard of carbon fixation or nitrogen fixation. It's a fancy term for just saying that you're taking the element from a form that's not usable and converting it into a form that the organism can use in order to do something with it, whether it's build cellular materials, get energy out, whatever it needs to do with it. You're converting it to that usable form. In order to do these processes of fixation, it does require ATP, it does require reducing. One of the ways by which organisms fix carbon is through something called the Calvin cycle, something I know you guys have heard about before. I see some of you shaking your head. You do not need to learn all the steps of the Calvin cycle, okay? The Calvin cycle is often taught in general biology as the second stage of photosynthesis, right? It's the stage by which carbons are fixed into molecules of glucose that the plants and other photosynthetic creatures can then use as their food, right, to make energy from through glycolysis and cellular respiration. So this is the very same Calvin cycle, also known as carbon fixation cycle. You're taking CO2, carbon dioxide from the atmosphere, and you're putting it in a more usable form for the microorganism. It's used by autotrophs, right, those photosynthetic creatures, including all the oxygenic phototropes. It's found in microorganisms such as the purple bacteria, the same ones we saw in that salt marsh in the previous slide, as well as the cyanos, algae, green plants, of course, but those don't really fall into microbiology, but we'll include them here. And most chemolithotrophic bacteria and a few archaea. So for the Calvin cycle, what do we require? Well, we require the CO2 from the atmosphere. That's not usable and needs to be made usable. And then we use a CO2 acceptor. That's something that's going to bind to the CO2 and accept it. NADPH, this should look a lot like the electron carriers we've been studying, right? NADH and FADH2. This one, though, has an extra phosphate group, so it's NADPH. Sometimes students tell me to remember this one because it has a P, and it's used in photosynthesis in the Calvin cycle. So that's one way you can remember it. But the P doesn't stand for photosynthesis. The P is an extra phosphate group in its chemical structure. But it's an electron carrier. And then you need ATP. Guys, remember, we are doing biosynthetic, anabolic, endergonic, non-spontaneous reactions here. We have to put energy, ATP, into these processes to make them happen. We've switched. Up until now, we've been talking about catabolism, where you get energy out, you make ATP. Now we have to put it in order to build cellular macromolecules. Now, in addition to those raw materials that I just mentioned, you also need two very important enzymes. One of them is rubisco. That stands for ribulose bisphosphate carboxylase. And this is a molecular model based on this crystal structure of rubisco on your slide. It's beautiful, isn't it? I love how they did the rainbow colors. This is thought to be the most abundant protein on our earth, rubisco. So the next time you're watching Jeopardy and questions asked, you'll know. So rubisco is well studied. We know the structure of it, and it's super important in the Calvin cycle. And then another enzyme that's important in the Calvin cycle is phosphorybulokinase. And so basically what can happen is the CO2 that was in the atmosphere in an unusable form can be made into a usable form like glucose sugar through this process of the Calvin cycle. And guess what glucose sugar can then go into? Glycolysis, right? And then maybe cellular respiration or fermentation, whatever the organism needs. So one way to consider the Calvin cycle is CO2 has one carbon in it, right? So you're taking a one carbon molecule from the air and you're fixing it into a 6 carbon glucose, or 6 carbon sugar called glucose, right? So you can think of using 6 CO2s to make one 6 carbon sugar. But it's a little more complicated than that. But that's sort of one way to think about it, right? You need 6 of these CO2s in order to create the 6 carbons that make up that sugar, C6H12O6. We call 6 carbon sugars hexoses, by the way. The hex means 6. So really what's happening, though, is you result, at the end of this, you get 6 molecules of something called ribulose 5-phosphate. That has 30 carbons in it, because these are 5 carbon sugars, and then you get your 1 hexose 6 carbon sugar that's used for glucose. And then that glucose can be used to build for biosynthetic reactions, right? You can use glucose monomers to connect them together with glycosidic linkages and make starches and cellulose and chitin and glycogen and all these carbohydrates that are used throughout the cell, right? Or it can go through glycolysis and get more energy out if you need that. Phosphoribulokinase is that enzyme, one of the enzymes I mentioned. It phosphorylates each of these ribulose 5-phosphates. And whenever you phosphorylate something, you're adding a really high energy bond there, right? So more ATP is needed to phosphorylate this. And you wind up regenerating ribulose bisphosphate, which is a 5 carbon sugar. So you have to invest quite a bit. In order to build this 6 carbon sugar, this hexose, and these 5 carbon sugars, you had to put in 12 NADPHs, okay? So that's, remember, NADPH, if it's got the H on the end, it's the reduced form. Does that mean it has electrons or doesn't? It has the electrons, right? And the electrons are energy. So NADPH has those high energy electrons. Those have to go into this system, and they have to invest those high energy electrons into the system because you're building sugars, right? Same with ATP. You have to invest. You have to put in 18 ATP to make one single 6-carbon sugar glucose. It's a lot of ATP. It's expensive. Glucose is expensive. And just keep in mind, this is just one way to fix carbon from the atmosphere into a form that cells can use, but there are alternative ways. All right, any questions about the Calvin cycle? Oh, I forgot, I have a figure here. You do not need to memorize all the steps of the Calvin cycle, okay? So make a note for yourself. If I mentioned different enzymes on the slides, like in the previous slide, then you probably want to know those. But as far as like here, you don't need to memorize all these steps. So if we start here, this is the CO2, which will be your raw material, and you're going to ultimately need 6 of them to go through this cycle to build one of these 6-carbon sugars. And they have fructose here because fructose actually comes off the cycle, and then it gets immediately made into glucose for biosynthetic reactions. So here's your CO2, and 6 of those are going to have to go through the cycle before you get your 6-carbon sugar. But also keep in mind that you need to invest ATP at this stage, again at this stage, and you need to invest high energy electrons at this stage in order to get that 6-carbon sugar for biosynthetic reactions. Okay, so that's carbon. We fixed carbon now, at least by one method. We're not going to go through all the methods. What about nitrogen? Nitrogen's super important, right? Where is nitrogen found? It's in all amino acids, right? Amino means nitrogen containing. Where else? Nucleic acids, good. Yep, nucleic acids have nitrogenous bases, A, C, G, T, and U, right? Nitrogenous, nitrogen containing. So we need nitrogen, absolutely, for an organism to be able to survive and thrive. So let's figure out how microorganisms will take nitrogen from the atmosphere in the form of N2 gas that surrounds us and makes it into an actual nitrogen-containing compound that they can use to build their own cellular macromolecules. Again, this process is known as nitrogen fixation, and usually what they'll do is they'll take N2 and they'll make ammonia, NH3, or nitrate, NO3, with a -1 charge. So this is kind of a complicated figure, and I don't really want you to know any great detail, but there are some parts that are super important, namely the enzymes. So if we start at the top here, you'll recognize pyruvate as being the product of glycolysis. So it can begin there at that step, but it can also begin at later steps as well. But when pyruvate gets oxidized to acetyl-CoA, electrons get released. And then those electrons are going to be transferred between this flavodoxin, and electrons are going to be transferred between this enzyme dinitrogenase reductase. Nitrogenase reductase, right? So electrons are going to be transferred. It's going to cycle between the reduced and oxidized form. And in order to reduce it, ATP is going to have to be used in this process. And then another enzyme, dinitrogenase, is also going to go through oxidation and reduction until ultimately N2 has been converted to ammonia, NH3. So if we look at the summation at the bottom, you can see N2 starts off with a nitrogen atom triple bonded to another nitrogen atom. That triple bond contains a ton of energy in those electrons. Triple bond, that's really stable. A lot of energy in there. You gotta break through that. So what does it do? It chops through it one at a time. So this triple bond goes to a double bond, right? Electrons get released and generate some hydrogens that get released. And then this one gets broken from a double bond into a single bond. Electrons get released, more hydrogens get made. Until ultimately this one gets broken and then you make 2 ammonias, right? So each time energy is getting released, hydrogens are getting made and released until you've ultimately converted into something that wasn't usable at all, into something that is super usable for the cell. So again, no need to know the details of this figure, but the key points are these enzymes that make up the nitrogenase enzyme complex. So you've got dinitrogenase reductase and dinitrogenase, and they're constantly cycling back and forth between their reduced and oxidized forms, depending on whether they're gaining or losing electrons. And ultimately, N2 gets converted into ammonia, NH3. So nitrogen fixation is catalyzed by this nitrogenase enzyme complex that I mentioned on the previous slide. It consists of two enzymes, dinitrogenase and dinitrogenase reductase. And those cycle between their reduced and oxidized forms. There's actually a cofactor associated with dinitrogenase. It's an iron molybdenum cofactor. Remember, cofactors are basically these inorganic molecules that are associated with enzymes that help them do their job better, right? And they often are tightly covalently bound in there. They're part of it. Think about like the heme group containing iron, right? And those cytochromes for hemoglobin. So this is an iron molybdenum cofactor, and it's really where the nitrogen reduction is occurring. That triple bond between the nitrogens and that N2 molecule is very stable, and it makes activation and reduction very energy demanding. So you have to really go in there, break those bonds, release energy in order to make those hydrogens. You actually need 6 electrons, and 8 are actually consumed because H2 is being produced. 16 ATP are required to lower the protein's reduction potential, enabling the enzyme, dinitrogenase reductase, to reduce dinitrogenase. Very energy intense reaction, but absolutely necessary, right? Proteins and nucleic acids cannot be built without nitrogen, and nitrogen fixation is absolutely necessary for microorganisms to be able to use the nitrogen in the atmosphere, which is their main source of nitrogen. So the nitrogenase enzyme complex is actually inhibited by oxygen. So in those organisms that do use oxygen, like the obligate aerobes, the enzyme winds up being protected from oxygen so that it can't keep it from doing its job well of nitrogen fixation. So there's a combination of things in place that keep oxygen away from the nitrogenase. Removal of oxygen by respiration, right? Whenever oxygen receives those electrons at the end of respiration, it gets converted into what? Water, right? Water is the byproduct of respiration. So you can constantly be removing the oxygen and making water by continuing this respiration pathway. There's also the production of oxygen-retarding slime layers that can help keep the nitrogenase free of oxygen. And the nitrogenase can be localized in particular structures known as heterocysts. And here's actually a fluorescent microscope image of a heterocyst that would house the nitrogenase. I don't exactly know how the nitrogenase knows to go there. I think it's really cool that there's a special structure in place to protect it. So that's carbon and nitrogen fixation, okay, which are so important for proteins and nucleic acids. Let's talk about sugars. You have to have sugars for a variety of reasons inside the cells of microorganisms. Can you think of any reasons why you need sugars? Energy, good. Ribose, absolutely. Ribose is a 5-carbon sugar, and it's part of RNA, and deoxyribose is part of DNA, and ribose is part of ATP. What else? Remember that? Yeah, the LPS layer of bacteria. Do you remember that? Lipopolysaccharide, polysaccharides, sugars, carbohydrates, right? When we learned about the cell walls and cell envelope of microbes. So polysaccharides are ultimately synthesized from activated forms of glucose inside microorganisms. The 2 types of activated glucose that we'll talk about are uridine diphosphoglucose, abbreviated UDPG, and adenosine diphosphoglucose, ADPG. UDPG is shown on the right here. You can see the structure of it. So it has your traditional glucose molecule here in blue. And then you have 2 phosphates, that's the diphosphate part. And then you have your ribose sugar and your uridine nitrogenous base. So this is a modified form of glucose, right? And it serves as a precursor to the structural polysaccharides that make up the LPS, so bacteria, the NAG and NAM. Do you guys remember that? So this is calling back some of our knowledge of the structure of the cell envelope. And then adenosine diphosphoglucose is another one. So basically these activated glucoses, these modified glucoses, are going to be what's added on to a polymer fragment to create long polysaccharides connected by a glycosidic linkages. And ultimately, inside your cells, what you can do is, our microorganisms can do, and we can do, gluconeogenesis. Have you guys heard of this pathway? So it's kind of, you can think of it in a way as the opposite of glycolysis. So whereas glycolysis takes sugar and breaks it down into 3-carbon pyruvates and makes ATP, right? Gluconeogenesis takes a 3-carbon sugar and builds a 6-carbon glucose. So it goes the opposite way. So if you remember PEP, or phosphoenol pyruvate, that was the intermediate in glycolysis right before pyruvate. So you start with PEP and then you can generate glucose going reverse. Now the reactions aren't exactly reverse, because some of those reactions are highly endergonic or exergonic, so you have to kind of do different reactions. But the ultimate reality, like the end product, is you start with the end of glycolysis and you get the beginning. Now, 6-carbon sugars like glucose, like we just talked about on the previous pathway, that can be assembled through gluconeogenesis using those activated glucose molecules. 6-carbon sugars aren't the only ones. Yeah, those are going to be the main ones used for energy for your cells. But 5-carbon sugars are also super important. You guys already mentioned ribose and deoxyribose, which make up nucleic acids. Those are 5-carbon sugars. And then ribose is also a component of ATP. So 5-carbon sugars have the name pentose, just like 6-carbon sugars are hexose. And they're formed from hexose by removing one of the carbons. And as I mentioned, they're required for synthesizing nucleic acids. Ribonucleotide reductase is actually an enzyme that can convert ribonucleotides to deoxyribonucleotides by just removing that oxygen. Now, how do you make pentoses? Well, the cells of microorganisms use a pathway for that. There's a pathway for everything in metabolism, isn't there? And this one's called the pentose phosphate pathway, or PPP for short. And it's an alternative to glycolysis. So basically, an intermediate in glycolysis can exit glycolysis and then enter the PPP instead and make 5-carbon sugars, as opposed to breaking it all the way down to pyruvates, which is 3-carbon. So glucose gets oxidized to CO2, NADPH, and ribulose 5-phosphate. And that ribulose 5-phosphate is a pentose that can then get converted into other pentoses, including ribose and deoxyribose. So we're basically making pentoses through this pathway, and the 4 to 7 carbon sugars that are produced include ribose, important for nucleic acids. Now, I think of the PPP as doing 2 things for the cell. Yeah, one of them is making ribose, from which you can also make deoxyribose for nucleic acids and ATP. But the second important thing that the PPP does is it makes NADPH. What is NADPH? A very important electron carrier, okay, that's used in different pathways like the Calvin cycle that we mentioned earlier. So you actually get NADPH from this pathway as well. And that's important for a variety of processes, like we said, the Calvin cycle, deoxyribonucleotide, and fatty acid biosynthesis. So this shows you how PPP works. It's 2 steps. The first step will take an intermediate from glycolysis that exits the glycolytic pathway and it's converted to ribulose 5-phosphate, releasing, making NADPH in the process, and releasing a CO2. The second step is to take that ribulose 5-phosphate and then convert it to a variety of different 5-carbon sugars. So you wind up making all these different 5-carbon sugars through this pathway. The most important of which being ribose, which is going to be important in nucleic acid and ATP synthesis. That's the PPP. What slide am I on? My alarm went off. How many more do I have? Oh, I got close, didn't I? All right. We will finish this next time. Don't leave yet. Is Trevor here? Trevor Wilson? Trevor? No. Ooh, that's right. Sorry, I should have warned you. All right, guys, so we're going to go over now some of the most missed questions from the exam. Overall, you guys did well. The average was a 73%. I aimed for a 75, so 73 is not too bad. Hopefully next time, though, now that you guys are used to how my tests are, hoping that average will be higher, okay? I also did throw out some questions that were missed by a lot of students, because that means that I either didn't teach it well, or maybe the wording was confusing, something wasn't right, so I threw out those questions, okay? Please do come by my office to take a look at your test if you haven't already. And you can spend as much time as you want with the test, okay? Please don't take any pictures. So the first part was multiple choice. I should mention the high grade was a 98 too. So congratulations on that. For the multiple choice, you guys did really well, especially on this first page. I was really happy that you guys were able to apply Coates postulates like this. That's what I mean by application-based questions, right? I'm not going to necessarily ask you which of the following is not part of Coates postulates. I'm going to ask you, okay, given this, how can you apply it, right? And you guys handled it very well. So this did well. These, though, did not go so well. So I pulled this figure from a lecture slide, and it showed you the growth of different microorganisms with respect to temperature. So I wanted you guys to see that right here between 30 and 40, this green curve has a maximum or optimum growth here at right around body temperature, right? So that would be your mesophils or mesophile, would be B. And then I wanted you to apply your knowledge of the proteins and membrane structure of different organisms with where they tend to live, right? So if you have enzymes with more alpha helices and more polar amino acids, what does that mean? Do they live in the hot or the cold? Cold. So that would be these that have the lower temperatures, A. And then what about ones that benefit the most from membranes with long chain, saturated fatty acids, fewer unsaturated? That means you have a very stable membrane. What would that be? All the way to the right, the hottest environment, right? The benefit the most. I was hoping for E here, okay? So when I do show you these figures in class, make sure you really understand them and you can apply them, okay? Come see me if you have any questions, of course. So those were, those were, I would say half of you guys probably got those, or maybe a little more than half. I think I did wind up throwing out one or two of those. And then, let's see, the next one was this one, chemolithotropes, right? These guys use inorganic compounds. We actually talked about that today, didn't we? 13, compared to eukaryotes, bacteria and archaea have blank surface to volume ratios, causing blank nutrient exchange. So this goes back to our discussion of how prokaryotes tend to have smaller cells. And if you have smaller cells, you have a smaller volume, right? So you have higher surface area to volume for the smaller cells. That's why cells are smaller, right? So they have more surface area for the amount of volume, and therefore they're able to exchange nutrients across that membrane better. Y'all did well on these. The next one that gave students trouble was number 21. I threw it out. By controlling the concentrations of nutrients continuously added in the chemostat, cells can constantly maintained at exponential growth, right? That exponential, that's when everything's working at a max. All the enzymes are working well, the cells are growing, right? That's when you want to do all of your experiments, and that's the point of a chemostat. You have cells at that stage all the time, right? Once you reach stationary growth, you have just as many cells dying as you do living, right? And you're not interested in that because you could take a sample out and get half dead cells. That doesn't help you, right? So really that exponential phase is what. Yeah, the rest of the multiple choice you guys did well on. But the fill in the blank was awful. I don't mean to laugh like funny, like I just mean that I'm not going to be doing fill in the blank, I don't think anymore, because when I saw this, I was like, oh no, I did not expect it to be tricky. Okay, so I will not be doing fill in the blank. Instead, I'll probably keep true false. You guys did well with true false, and I'll just do maybe one extra short answer. But this really surprised me. I did not mean for this to be as hard as it was. So I did wind up throwing out, I think, 2 or 3 of these. So this first one is talking about resolution. That's when you're looking at a microscope and you can't distinguish 2 things. They sort of merge together. That's because of resolution. This one's the energy source derived from the charge difference with respect to hydrogen ions across the membrane. That's your PMF. I think you guys knew what I was talking about, but you couldn't think of the exact term. And I didn't anticipate that. Now I know. This is the endosymbiotic theory. The time interval for the formation of 2 cells from 1 generation time. Acidophiles grow best at low pH. Anaerobic organisms cannot respire oxygen. Okay, the short answer went well. You guys did great. The short answer, I was super happy. And I love being able to give you guys partial credit because I can see where you're going. Like, oh yeah, you're almost there. But you didn't mention this really important thing, so I've got to take off a point. So I wanted you guys to tell me about Pasteur and some of the contributions he made. Many of you used the Swan Neck flask, and it wasn't enough just to say Swan Neck flask because I just gave that to you. You needed to tell me what that was and how it disproves spontaneous generation and all that stuff. So I was looking for that, or you could have talked about alcoholic fermentation being a biological process. Living organisms discriminate between optical isomers, sterilization, food pasteurization, vaccines, any of those would have been fine. Again, Swan Neck flask, I wanted just a brief 1-sentence description of how it was done. Basically, that when he bent the neck, right, he was able to keep the microbes out and it was sterile, but then when he poured it over and the microbes came in contact with the media in there, you did get microbes, and it disproves spontaneous generation. For this one, I wanted you guys just to mention either membrane or wall, some differences in the chemical composition between bacteria and archaea. So for bacteria, for instance, in their membrane, they have what kind of hydrocarbons? So their phospholipids are made up of what? Fatty acids, good. Do archaea have fatty acids? No, so that could have been one difference, right? Archaea have isoprene. Remember that? Also, the linkages there, bacteria have ester linkages, whereas archaea have ether. And then bacteria are always bilayer, lipid bilayers, but archaea are sometimes monolayers. So those are 3 right there with the membrane. With the cell walls, you could have talked about peptidoglycan in bacteria, no peptidoglycan in archaea. They talked about differences in the sugar backbones for the LPS, NAG, NAM for one, and then the other one has no NAM, it has another variety. The linkages are different, beta-1,4, beta-1,3. L or D isomers, just L isomers for the amino acids, can be destroyed by lysozyme and penicillin, cannot. May have a polysaccharide wall, doesn't have an S layer. Any of those things would have been fine, okay? So just don't ever leave a question blank like this, okay? I gave credit to all kinds of things, okay? But if you wrote nothing, you got -8. And that's a lot of points, okay? So write something. I can make some sense of most things, okay? And then the advantage would be they can live in harsh environments. And then for this one, I just wanted you guys to draw the curve. So you would start out sort of low, and there'd be a lag there, right? So you might have missed points if you went straight up. because there's a lag phase, right? And then you have exponential phase where it goes up linearly. And then it plateaus, stationary, and then it goes down. That's it. So I'll see you guys on Thursday then.